Today, my talk will focus on the latest offerings from Crown Bioscience, that is to harness high-quality in vitro screening using tumor organoids, complemented by high-content imaging technology. While there are many definitions for the word organoid in the scientific field, at Crown Bioscience, we use the term organoid specifically to describe the technology established by Hans Cleaver's lab and further developed at Hubert Organoid Technology as hub organoids. Now, these organoids are derived from LGR5 positive adult stem cells of epithelial origin. Expansion and differentiation of these stem cells gives rise to functional mini organs in a dish that can recapitulate the genomic, morphogenic, and physiologic characteristics of corresponding organs in vivo. The hub organoid technology does not require reprogramming or transformation of stem cells, and therefore enables the development of diseased as well as healthy organoids, which preserve the original tissue genetic and epigenetic makeup, including clinically relevant mutations. At Crown Bioscience, our 3D in vitro platform can be categorized into 4 basic types based on the cell types. As one, the 3D tumoroids. Now, these are derived from our CDX or cell line derived xenografts and PDX cell lines. The advantage of using this is that the turnaround time is really quick. The second one is the PDX derived organoids, also called as PDXO, and as mentioned, they are derived from our PDX tissues harvested from mice. The advantage of using this platform is that they have matching established in vivo PDX models already at Crown Bioscience. The third one is the PDO, or patient-derived organoids. Now, these are derived directly from patient tissue and have never been passed through mice. When available, these models have primary and metastatic pairs, as well as normal and tumor-matched pairs. The fourth one is the 3D EVPT platform, or the ex vivo patient tissue platform from fresh tumors, which gets processed within 24 hours of tissue resection. Now, this is a one-shot assay for a given patient, wherein the native tumor microenvironment is preserved. This assay is suitable for late-stage clinical evaluation. As a service provider, Crown Bioscience has generated a living biobank of organoids. Currently, there are 390 PDXOs and 216 PDOs available for moving into studies. These organoid models cover a variety of indications such as breast, ovarian, pancreatic, colorectal, and lung. Matching tumor and organoid, normal organoid pairs, organoid and PDX pairs for follow-on in vivo studies are also available. These models have been genetically characterized by RNA sequencing and whole exome sequencing, and their response to common standard of care agents as well has been characterized. The next obvious question is how, why, and what makes Crown Bioscience organoids unique for drug screening programs? One, the organoid platform is the only in vitro platform using clinically relevant models for high throughput screening and are clinically predictive to patient response. Two, they are easily scalable to generate biobanks, test multiple models, and drugs simultaneously. Three, they have high superior efficiency with high signal to noise ratio, high reproducibility, and low batch-to-batch variation. And lastly, they have matched in vivo models to bridge the transition from in vitro to in vivo. Now let's discuss briefly on the study format and readout. All organoid screening studies are carried out in 384-well microtiter plate format with automated plating, treatment, and plate readouts for survival and for apoptosis. Cell viability readout using cell titer glow or CTG is the most common and cost-effective readout method. In addition to CTG, we also provide high-content imaging readouts that provide more detailed insight to understand complex disease pathophysiology. HCI enables cellular level as well as temporal and spatial analysis to understand delicate intracellular distribution, localization, cell morphology, and other changes that occur upon treatment. A typical high-content imaging study begins with seeding 100 to 500 organoids per well, followed by treatment and incubation for typically 7 days, fixation, staining, and imaging by Z-stack optical sectioning. The 3D images are then reconstituted or reconstructed and using multiple parameters such as cell size, shape, number of nuclei, etc. The effect of the drug exposure is phenotypically profiled. The outputs can be varied. It can be IC50, tumor and nuclei count, the size of the nuclei, the size of the cell, the volume, shape, necrosis, internalization, and even localization. For more information on the organoid models, please visit our organoid database. This is a free database and only requires registration. Data on IC50 values, light microscopy, histopathology, genomic data, and patient data are all available. I'll now proceed to show you a case study that used the organoid platform for antibody screening. This paper was published last year in April in Nature Cancer and was a joint collaboration of Suprastem and Crown, which was at that time Ocello. The Suprastem consortium consists of Urs as the coordinator, the Hubert Institute in Netherlands, Ocello, Institute of Research in Biomedicine, Barcelona, Spain, and the Wellcome Trust Sanger Institute at UK. This manuscript describes a large-scale functional screen of dual-targeting bispecific antibodies on a heterogeneous colorectal cancer PDO biobank and paired healthy colonic mucosa samples. To give you a brief background and rationale for the study, it focuses on targeting the tumorigenic potential of cancer stem cells in colorectal cancer. These cancer stem cells are characterized by elevated level of Wnt receptor complex, including LGR5 and the zinc finger proteins. The antibodies were designed to target the Wnt pathway while also simultaneously targeting tumor growth and survival driven by the receptor tyrokinase pathway and the EGFR pathways. The goal of the paper was to develop a therapeutic strategy based on blocking proliferation of cancer stem cells in colorectal cancer by leveraging the dual targeting capabilities of the bispecific antibodies, and also generating and use of CRC patient-derived organoids or PDOs biobanks to functionally screen and discover novel drug candidates. More than 500 therapeutic bispecific antibodies were generated against the Wnt and the RTK targets by Urs using the MIMO transgenic mice, as shown here in this slide. Meanwhile, Ocello or Crown Bio generated 99 CRC organoid models from 68 patients with 32 tumor-adjacent healthy organoids. These PDOs were characterized for their morphology and mutational status. The 500 plus therapeutic bispecific antibody candidates were screened against the CRC PDOs of multiple genotype and phenotypes to assess their functional activity and compared to matched healthy colon mucosa organoids, as shown here in the two panels. Analysis was based on high contact imaging to capture the complexity of PDO responses. For this approach, they identified MCLA-158, LRG5, and EGFR bispecific antibody with potent selective growth inhibitory activity. This antibody specifically targeted EGFR degradation in LGR5 positive cancer stem cells, but showed minimal toxicity towards healthy LGR5 positive colon stem cells, as shown here in this graph. The results from the in vitro studies were then further confirmed in vivo using CRC PDX models. This paper described for the first time the use of PDO biobank in a drug discovery pipeline. The bispecific antibody is currently in clinical trial. For immuno-oncology testing, using the same 384 well format, organoids can be co-cultured with immune cells such as PBMCs or isolated macrophages or T cells. Now, the IO test agent can then be introduced into the wells to determine sensitivity, and the readout is always based on HCI. Assays such as migration, aggregation, killing, and infiltration can be evaluated for multiple therapeutics such as CAR-Ts, ADCCs, and ADCP. On this slide is an example for treatment-induced immune cell migration, showing movement of cell tracker-stained immune cells from the top of the hydrogel towards the tumor cells located in the lower half of the well. This is a side projection image of the well. Image analysis is done to score the number of immune cells in different Z positions for the evaluation. An example for model-dependent sensitivity to killing is shown in this slide. For this, 5 colorectal cancer organoid models with different morphologic characteristics were co-cultured with naive PBMCs, all from the same donor. The PDOs showed different sensitivity to immune cell-mediated killing. While all models showed a mild killing upon addition of the naive PBMCs, the stimulation with the superantigen resulted in killing of the organoids from 30 to 90%. Next is an example for immune cell donor-dependent sensitivity of killing. Co-culture of one CRC model with 15 different PBMC donors is shown here. While all the donor PBMCs showed consistent killing, stimulation with superantigen enhanced killing effects by 50 to 80%. And on this slide is an example of myeloid cell analysis in co-culture. Analysis here was based on multi-feature scoring on parameters such as shape, size, and spikiness. It showed that the M1 macrophages had the highest infiltration into the tumor organoids compared to CD14 positive monocytes or the M2 macrophages. Similarly, triple co-culture experiments of organoids with CAF cells and immune cells can also be performed to evaluate tumor microenvironmental changes. In summary, The combined power of organoids and high-content imaging offers a higher level of in vitro screening that is physiologically relevant, can be automated into 384-well plates, including readouts, superior image analysis to measure phenotypic changes, and is also reliable and the data is reproducible. Thank you.